Hey Satterton, this is Mr. Dubik, and in this video we're going to be talking about the first two stages of cell respiration, which is glycolysis and the prep stage. Alright, so the first stage of cell respiration is called glycolysis, and if you break the word down into glycolysis, you find two words: glyco, which means glucose or sugar, and lysis, which means to split. So in this process of glycolysis, literally what we're doing is we are splitting a glucose molecule. So hopefully that helps you out. Now, what organisms go through glycolysis in cell respiration? And glycolysis is used by all living things. And why is because it is anaerobic. So glycolysis can occur whether oxygen is present or not. And so therefore plants, animals, and bacteria can all go through glycolysis. Okay, so where does glycolysis occur? Glycolysis occurs in the cytoplasm of the cell. So this process right here, this first stage of cell respiration, takes place outside of the mitochondria. So again, it takes place in the cytoplasm of the cell. This stage of cell respiration is anaerobic, which means oxygen does not have to be present in order for this process to occur. So again, oxygen is not required in order for glycolysis to happen. What actually is going on during glycolysis? And again, this is a nice overview right here of what glycolysis is and what happens during glycolysis. And that is one glucose molecule, which comes from the food we eat and photosynthesis, is broken down into two molecules of pyruvic acid or pyruvate. So again, in glycolysis, one glucose molecule is broken down into two molecules of pyruvate. And again, we'll go into that process more in the next couple of slides. And again, like I said before, glycolysis, if you break the word down, glycolysis, that means that it is the breaking down of a sugar. So here is the overall process of glycolysis. To the left, you see this diagram of glycolysis, and to the right, we're going to talk about the reactants, products, and what really happens after glycolysis. So again, here I'm going to talk about the diagram first. So in glycolysis, we're starting with a glucose molecule, and a glucose molecule, if you see up here, is a 6-carbon sugar. So 1, 2, 3, 4, 5, 6. So those circles are carbons. So that tells us that that is a glucose molecule. And so in glycolysis, we start with a glucose molecule, which is a 6-carbon sugar. And then essentially what we're trying to do is, if you look down here, we're trying to break it down into 2 3-carbon molecules of pyruvate. Alright, and so we're just going to go simply down this diagram and explain that. So we're going to start with a 6-carbon molecule of glucose, and then what's going to happen is we're going to add these phosphates to make this glucose molecule a little bit unstable so that we can split it in half. So again, glucose molecule is going to add 2 phosphates to it to allow it to become unstable so that we can split it. So then what's going to happen is we're going to add 2 ATP molecules to this process to help split this glucose molecule. So phosphates are added, ATP is added. Now the glucose molecule splits into 2 3-carbon molecules called glyceraldehyde, which still have phosphates attached to it, and we need to make sure that these phosphates are off so that we can get pyruvate. So how do we get these phosphates broken off? Is what we're going to do is we're going to break these phosphates by adding NAD+, which is going to essentially free off electrons from the breaking of this bond. Those electrons are going to be added to NAD+. The NAD+ is going to accept those electrons, and then as a result, it's going to add a hydrogen to it to form NADH, which is important. Okay, now in the formation of these 2 3 molecules of pyruvate, what is essentially happening is we're gaining 2 ATP molecules per pyruvate. So essentially we're yielding 4 ATP molecules at the end of this process. However, we had to use 2 ATP molecules to split this 6-carbon molecule of glucose into 2 molecules of pyruvate. All right. So essentially in glycolysis, we start with a glucose molecule. We use 2 molecules of ATP to split it into 2 molecules of glyceraldehyde, which still contain phosphates. We have to remove these phosphates. So we're going to use NAD+, which is going to help break off those phosphates. Essentially, then what we get is we get NADH. And then for every pyruvate that is produced at the end of glycolysis, we're going to net 2 ATPs for a total of 4. Therefore, we end up with 2 pyruvic acids, 4 ATPs, but again, we use 2 ATP molecules up here to split glucose, so we only have a net gain of 2 ATP molecules. And then also for each pyruvate that is produced, we're going to form and produce 1 NADH. So for a total of 2 NADH molecules. So if oxygen is present in the environment, we're going to go through aerobic respiration. If no oxygen is present, we're going to go through fermentation. So in this presentation, we're going to go through an aerobic environment and then move on to the prep stage. Alright, so we just got done talking about glycolysis, and remember, glycolysis, we start with 1 glucose molecule and split it into 2 molecules of pyruvate. Those molecules of pyruvate consist of 3 carbons. And at the end of glycolysis, what do we produce? We produce 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. So now what we're going to do is we're going to continue on in cell respiration, and we are going to go to the prep stage. So now we're going to enter the mitochondria. So what we're going to do now is we're going to talk about what's going to happen to these 2 molecules of pyruvate. Okay, so now what we're going to do is we're going to talk about stage 2 of cell respiration, which is called the prep stage or the prep reaction. So essentially in the prep reaction of cell respiration, what's going to happen is the pyruvate produced in glycolysis is going to be made into acetyl-CoA using coenzyme A. And we'll explain that process in a minute. And then as a result of this little reaction going on, NADH and carbon dioxide are going to be released and produced as byproducts. So again, pyruvate is made into acetyl-CoA using coenzyme, and as a result of this reaction, NADH and carbon dioxide are going to be released and produced as byproducts. So if you look at this diagram down below, you can see that our starting product is pyruvic acid. Pyruvic acid is now entering the mitochondria, okay? And essentially what's going to happen to this pyruvic acid is it's going to be broken down using coenzyme A to form acetyl-CoA. That's literally all the prep stage is. Pyruvic acid is going to bond with coenzyme A to form acetyl-CoA. Now, how does it do that? So pyruvic acid, which is a 3-carbon molecule in the mitochondria, is going to bind with coenzyme A. When coenzyme A binds with pyruvic acid, a single carbon is going to be lost. And that carbon right here is this group right here that's highlighted in blue. It's called a carboxyl group. And if you look at it, it's CO2. So what you're actually seeing here is the releasing of this 3rd carbon is actually releasing a carboxyl group. And essentially what's being released from this reaction is carbon dioxide. So this is a carbon dioxide molecule that is leaving, and that's actually what we exhale. Okay, so pyruvic acid enters the mitochondria, it bonds with coenzyme A to form acetyl-CoA. And now acetyl-CoA is going to be a 2-carbon molecule bonded to coenzyme A. And essentially we want to get acetyl-CoA because acetyl-CoA is needed to enter the Krebs cycle in order for it to work and function. Pyruvic acid cannot enter the Krebs cycle. And so that's why we have to use coenzyme A to bond to pyruvic acid to form acetyl-CoA. Now in the process of the breaking off of this 3rd carbon, a bond is going to be broken, so therefore electrons are going to be lost. And again, our partner NAD is going to come into the picture. It's going to pick up those lost electrons, and as it does that, it's going to be reduced from NAD+ to NADH. So NAD+ is going to pick up those lost electrons, and as it does that, it's going to also pick up a hydrogen, and NAD is therefore going to then be able to go to the electron transport chain. So essentially that is the prep stage of cell respiration, and again it is needed because we need to break this pyruvic acid down to form acetyl-CoA so that it can enter the Krebs cycle so that the Krebs cycle can function properly. Okay, so on this slide, what we're going to do is we're just going to review the first two stages of cell respiration, which is glycolysis and the prep stage. Okay, so remember, the first stage in cell respiration is glycolysis. Glycolysis occurs in the cytoplasm of the cell. And in glycolysis, we are taking one glucose molecule, which is a 6-carbon molecule, and we are splitting it into two molecules of pyruvate. And these pyruvate molecules consist of 3 carbons. Also, in glycolysis, we're producing 2 ATP molecules and 2 NADH molecules, which are going to carry electrons and hydrogens to the electron transport chain. Now, after we go ahead and split this glucose molecule into 2 molecules of pyruvate, these molecules of pyruvate will enter the intermediate stage, or the prep stage of cell respiration, and will now enter the mitochondria. Each pyruvate will bond with coenzyme A to form acetyl-CoA, and then once acetyl-CoA is formed, it can then enter the Krebs cycle. The Krebs cycle and the electron transport chain is what we will talk about tomorrow. Okay, so that is all I have. We will continue cell respiration throughout the remainder of the week and review next week. If you have any questions, email me, and I will talk to you soon. Bye.